Thanks everybody for joining our call today for our webinar. I'm Mike Morrow. I'm one of the salespeople here at CDI Labs. CDI was formed about 10 years ago, a little over 10 years ago. We have sites in Baltimore, Mayagüez, Puerto Rico, and Toronto. And so we offer both services and also some of the products to be able to use in your own labs that Tyler will be describing in detail, and along with some of the applications that our customers use the products for. And so he's got his title slide up, and with that, I'll let Tyler go ahead and introduce himself.
Yeah, do you want, I guess maybe speak to the audience and how this came about, and then I'll kick it off.
Sure. So we had reached out, and Damien had asked if we would be able to do a webinar to present to the broader research community at UPenn, and that's how this came about.
Yeah, that's great. Well, thank you very much for the introduction, Mike. So for those of you that don't know me, which I assume is pretty much everyone on this call, my name is Tyler Hulett. My background is in cancer immunotherapy, immuno-oncology. I worked with Bernie Fox at the Providence Portland Cancer Center on cancer vaccines and watching immune responses to those. So I'm very familiar with kind of the IO space. Since joining CDI 6 years ago, obviously I've transitioned to a bit more general take on autoimmunity, which I'm going to try to impart some of the things that I've experienced or learned over looking at tens of thousands of samples for a lot of different types of projects, for everything from lupus to cancer immunotherapy to schizophrenia, bipolar disorder. We've looked at a lot of really interesting stuff. So the title of this talk, which has been streamlined by Mike and Damien, is that when immunity misfires, right, leveraging this type of self-recognition and these failures, and what is it that we can learn from this? So with that, I guess I'll go ahead and kick it off. So you're probably familiar with the general idea. There's only a few things that we test for in the clinic, your Roe and Law types of your anti-double-stranded DNA, the types of things that we look for in the clinic as regards to autoimmunity are not all that many different types of things. And I guess I'd like to kind of challenge you to think about this in an alternative way, that perhaps autoimmunity is actually relatively common, very individualized and stable with time. Which is a little bit alternative to that. And so why do I claim this is because I've looked at a lot of data from HuProt microarrays. So these are things that are out of our founding labs at Johns Hopkins nearly 20 years ago now. But the way that we make a HuProt chip, so it's essentially a human proteome on a slide. We have about 21,000 full-length open reading frames, 21,000 different yeast clones, each of which is expressing a full-length human protein. These are then purified by GST tags printed with a microarray printer, and we're able to come back and stain with patient serum, which obviously has many different types of isotypes, which then bind to the array. We come back with a fluorescent secondary, an old GenePix 4000B microarray scanner, and so then you're able to get physical data. So this is what essentially a HuProt experiment looks like. These are negative background stains, so showing what the secondary only does. You see kind of the histograms all clustering off to the left side, not a lot of positive signal. Whereas if everything's positive, this is our anti-GST control stain, you see that you get this big, broad range of proteins. So everything on a slide lights up. And what you get for an individual patient typically looks something like this. So this is before and after 5 years of aging for a single individual. This is an anti-IgG3 stain, so we're looking at a subset of the IgG responses. And most of our data looks like this. Day in, day out, if you run technical replicates or you run one person across a time course, what you'll generally see is a plot that looks a lot like this. So the majority of the signal that you see here is negative. This is a log scale, so the stuff that's up here to the top right is quite bright. So every person, depending on where you're setting your thresholds, are going to get between about 100 and 400 responses on a HuProt array. And these are really stable with time and really unique to them. And we'll come back to this a little bit later with some identical twins data. But just as kind of a starting off point is if you look pre-post at HuProt data, it's going to look a lot like this. You're going to have a few hundred autoantibodies that people are reactive to. The majority of those are to intracellular proteins. So you can imagine that these are, people will call these natural autoantibodies at times, like they're probably helping with cellular debris clearance and other things, and they're probably not directly communicating or contributing themselves to autoimmunity because they can't really affect things on the inside of the cell. But there are ones that bind extracellular and signaling proteins, and I do think, there is a lot of evidence that these affect results. This particular sample is before and after the COVID-19 pandemic, and so we have highlighted the COVID spike protein here. You see a whopping new response from 0 at baseline. You generally see that the original responses don't go away. There's nothing here in the bottom right quadrant, right? So immune memory is either stable or slightly building with time. And so this is very typical of what we'll look at across HuProt time course data, is that the types of autoimmune mistakes that we can observe are very unique, very stable, and slightly increasing with time. And I'll show this much more thoroughly later on, but I just kind of wanted to frame of, the types of things that we're talking about. So now each of these dots, of course, is a unique human protein. So what does and what can that mean, which is kind of the purpose of this talk. And so the general paradigm, right, would be that autoimmunity is relatively rare. I'm going to challenge that and try to claim that autoimmunity is relatively common, and that you perhaps should care about this. And why should you care about this? So one way is when we think about, what are the types of really large platform data that affect patient outcomes. Obviously, DNA changes affects that. So there's a lot of literature on, inborn errors of immunity, people having, different cytokine or receptor profiles, different MHC types. You're going to have different types of immune responses. Same way that you can have DNA creates a functional protein, mutated DNA can create a dysfunctional protein. Well, antibodies can do that too, right? And there's a really emerging and interesting literature on this, that you can be a genetically normal person, but acquire an autoantibody event, similar to what I'm kind of showing here, that then alters the way that the body behaves for the rest of that patient's life. And so I personally, and a lot of our clients that work with us here at CDI, think that this is perhaps where certain chronic diseases may be occurring. And so that's what we try to do. So I would encourage you to think that anything that a drug can do that's a monoclonal can also be caused by a naturally occurring autoantibody. Now, the odds of you walking around with something that is behaving exactly like an anti-PD-1 or anti-TNF are relatively rare, but there is a lot of evidence that we do acquire these types of things, and they occur more often than you might expect. So we work with several types of people that are mining elite responder autoantibodies for naturally occurring drug-like things like this. And some of those are actually being taken into the clinic. So kind of another layer of complexity to this, right, is so just because something is binding doesn't really tell us what it does. And so that's kind of part of our challenge, I guess, for us at CDI and for autoantibody seromics as a field, is that just proving that an antibody sticks to something doesn't prove if it's an agonist or an antagonist, if it's interfering with ligand binding. There's a lot of different types of things that an autoantibody can do, and really just discovering that interaction happens is really just kind of the first step in a longer process. So some of the literature that's, I think, is really paving the way of where this is going. So this is a really nice review by Jean-Laurent Casanova at Rockefeller, as well as Paul Bastard, where they go ahead and they catalog a lot of stuff. So there's a lot of groups like Steve Holland's group at NIAID that are going through and showing that naturally occurring anti-cytokine autoantibodies affect who does and who does not get certain types of infectious disease. So this is a kind of a profound thing. So they got into this because Professor Casanova's background is in genetic inborn errors of immunity, and they were finding people that were having similar types of effects, were acting as if they had a knockout to something, but they didn't. And what's been found is that there's several types of different immune modifying or immune function modifying autoantibodies that are being found. And my guess is that this extends not just to the cytokines, but probably to every type of surface and extracellular protein that exists in the human body. And so there are some nice clear examples of this being found, because immunologists can see very clear effects of anti-immune modifying antibodies. But really anything that's outside a cell is something that might be affected. And that's part of why I think this field is so interesting. So we're all walking around with some sort of number of acquired autoantibody traits. We don't know what many of these mean, but when we find them, they could potentially lead to actionable therapies. So one example of this is a really nice paper out of UCSF last summer, where they used a human phage display immunoprecipitation sequencing technology. They actually ran this themselves, but we run this exact library as well. And so what this is a bacteriophage display system where you have these long peptides on the surface of bacteriophages representing the entire human proteome. So about 700,000 different little 49-mer peptides. And they very, very clearly showed with lots of validation assays that there are some amount of people, they originally found this in a single woman that was having CNS symptoms. They couldn't figure out what was going on. Well, it turns out that this woman and other people like them, they're actually seeing this in as many as 5% of the American population to some degree, is that there's a single section of a protein called transcobalamin or CD320. So transcobalamin is a receptor that is responsible for importing vitamin B12. So patients have normal circulating vitamin B12 in their plasma and serum, but it doesn't get inside cells. And it turns out that over the long term, this results in CNS symptoms. So you have an effect that kind of looks like this. So off to the left, we have, your normal B12 goes in through transcobalamin. We have healthy, happy, metabolically active cells. When this is blocked, you start to get all sorts of oxidative stress. We don't have the appropriate cofactor that cells need to be fit and healthy. And this probably creates its own interesting antigen set, which would be entirely secondary to the initial effect. So you can have these really complex downstream symptoms from a relatively simple perturbation. And I imagine there's a lot more stuff like this out there than has been discovered. And I think these keystone examples are kind of the signpost that there's a lot yet to be found in this type of space. So to kind of talk about autoimmunity and why we, I guess, traditionally have a gut instinct that it is rare, because it is rare. The body does not want to attack itself. This dates all the way back to Paul Ehrlich at the turn of the 20th century, where he coined a term called horror autotoxicus, or essentially a fear of autotoxicity. So there's a principle that the immune system does not want to attack the self. And he was able to do this with some of the first experiments that essentially showed that adaptive immunity existed, how it worked. They were able to show that there's heat labile components called complement that were independent from the memory component or the antibody. And he drew a diagram of antibody complement in a target cell that looked like this. And what they found was that if you immunized animals with the blood of other animals, they would generate a memory antibody response that could then kill or lyse blood cells. And they found that generally this did not occur within species, and very generally it could not occur within single individuals. And so he thought that there was this general principle, which there of course is, that the immune system is trying not to attack itself. Originally, they thought this was a law, right? They thought that autoimmunity was actually inherently impossible, and that events like this didn't really exist. So the title of this talk would be an immune misfire. So a literal misfire, so a literal anti-self response is what Ehrlich was thinking was impossible. The first experiments that really showed that this was possible were coming about in the 1920s. So there were people that were noticing that there would be really rare events after rabies vaccination where people would start getting this really horrible downstream paralysis. And so this was investigated. They went originally because of rabies and how rabies works. Obviously, it's a fatal condition, and so some people would think that, oh well, maybe there was still active rabies virus that was in the vaccination, and that's what resulted in these shots. This experiment over on the right, they were able to show that just by injecting adjuvant and rabbit brain, you were able to induce this effect. So they were able to show that it was entirely independent of the rabies virus itself, just by having adjuvant plus kind of a local mimic material. You could actually break tolerance. And we now call this Guillain-Barré syndrome. So obviously every immune response, whether it be a rabies vaccine or a modern vaccine, has some really low rate of something like this occurring, which is why if you go and you watch your ads for Shingrix on TV, the shingles vaccine, they'll actually note that there is a very small risk of Guillain-Barré syndrome, which can occur after this treatment. And things are, of course, a little more complicated than that. There's a really nice paper that just came out a few weeks ago showing that people getting their Shingrix vaccinations in the UK have lower rates of dementia long-term. And so, there's kind of a yin and yang to immunity. And one of our challenges as immunologists is trying to figure out, okay, when is the upside outweighing the downside? And I really think that we're really just in the earliest days of understanding when, where, and how this is always occurring. But obviously, we've done a good job with preventing quite a few diseases with vaccines. So this would be an accidental misfire, right? Some sort of injury, adjuvant, you get a literal break in tolerance. Another way that you can get effects like this is via molecular mimicry. So having, a structure of a virus such as Epstein-Barr virus that looks a lot like a self-protein. This is kind of more of an unattended consequence of normal immune behavior. Like what you really kind of have here is a bispecific to some degree. It's going to bind a natural protein. It's going to bind EBV, whether instead of just necessarily one or the other. So molecular mimicry as a concept really is about as old as immunology itself. So the original discoveries by Jenner on smallpox vaccination, cowpox is not smallpox, but there's enough molecular similarity to provide cross protection, right? And these types of anti-pathogen responses, while it's nice when they recognize similar viral strains and they help protect us from downstream similar infections, at times these proteins also look like our body's own natural proteins. So one really nice example of this that we were involved with, so this is out of Tobias Land's and Bill Robinson's groups at Stanford. And what they really showed very clearly is there's a subset of multiple sclerosis patients, about 25% of them, where they have an autoantibody that's detecting this one exact event. And so this is a molecular mimic between Epstein-Barr virus, EBNA1, and this human protein called gliocam. And that gliocam EBNA cross-reaction was found using a human protein microarray, a HuProt, which we make here at CDI with an isolated monoclonal. And they also ran VirScan FIPSeq. So we run this assay too, but this is one they did themselves. And they were able to also show that just the injection of the EBNA protein with adjuvant into mice could kind of recreate this anti-brain phenotype. So they didn't need to use the literal human sequence or the mouse sequence to induce this. They could just inject the EBNA sequence and get this anti-brain reaction. Some more recent work that we've been involved in with Akiko Iwasaki's group with long COVID, they've gone through and showed that people that, there's debate among the general public, right? Like, is long COVID a real disease? Is it in people's heads? Well, the people that claim to have these symptoms, they have autoantibodies to this protein called MED20 on a human protein microarray. You can inject human serum into mice and it will go and bind and stain to the mouse neurons. They've gone through and validated this both by ELISA and mass spec. And so it's kind of another example of you can have a chronic symptom with a literal molecular cause in immunity. And this is an example of that being found. And we really have one kind of final way that autoimmunity can occur. And this is by the body just changing itself with time. And so this part has probably been the least studied just because of all the ways that this can happen. But transformations to the body are also ways that autoimmunity can occur, both either by genetic mutation or by some sort of chemical transformation. Actually, the Epstein-Barr virus and gliocam reaction that I was just showing previously, it's actually a phosphorylated form of human gliocam that is the more dominant antigen in those cases. And so my guess is that a lot of times when we're seeing autoimmunity, say on a HuProt, perhaps that's via epitope spreading from some sort of more dominant transformation that's actually going on in the body. So anything that transforms a protein from what the immune system considers or expects to be self could, of course, also stimulate an immune reaction. And so healthy young human cells and proteins, the immunity is trained to not attack this, but it's kind of unavoidable to some degree that this is actually going to change with time. So one really nice lecture that's by Christopher Walsh that I've in the NIH Director Series, you can go and see this on YouTube. It's a really nice talk where they go through and are showing in non-dividing somatic cells like neurons, even those are requiring massively parallel genetic drift with time. And so they go through and are able to actually isolate this onto the DNA mismatch repair enzymes, where they've got like a window where they're correct, but if 2 mutations are occurring in some small zone, just natural mismatch repair and access of DNA is going to end up introducing mutations, and quite a few. So every neuron in your body is gaining in a very linear fashion about 17 to 18 single SNPs per year, and 2 to 3 indels. So this is a lot of genetic drift that's going on. And he goes so far as to claim that there's more genetic diversity within your body right now than in the intended somatic diversity of, just say, the parent gametes for every person alive on Earth. So immunity, being able to tolerate that, is obviously a challenge that is increasingly difficult with time. So going back a little bit to just genetic drift and evolution itself. So here's a diagram from Darwin's original On the Origin of Species. You have the parent organism here. We have all possible, variants that are existing today and extinct varieties along the way. What I'd encourage you to think about is that this is unfortunately simultaneously going on in the human body, both from genetic drift as well as acquired chemical insults. Like, there's quite a few things. There's just a paper. There's quite a few links between things like pesticides and neurodegenerative diseases. There's just a paper that came out a few days ago that was kind of making the public press on people living next to golf courses having a higher incidence of Parkinson's disease. And it's probably due to non-dividing neurons acquiring damage with time. This probably could lead to transformation that then leads to immune attack. And so what you want to think about with the aged body is that immunity's got a really tough go of things here, where the goal is to decide on what to tolerize against, but the body's continuously changing, and obviously it has to make some decisions about what it considers self and non-self, and those decisions are not always necessarily in our favor. And so, in spite of this, there do seem to be quite a degree of stability that's occurring with this autoimmunity across time, which we'll kind of get into a little bit. And so what we think, and with a lot of people that work with us think, is that immunity is this selective force that can alter health outcomes, and that these can be kind of permanently skewing, disease and health behavior across time. So one way that Darwin thought about this, at least with regards to unicellular organisms, this is a quote about animals that cannot interbreed, and by that he meant like single cell organisms like living in ponds. And with respect to organic beings which do not propagate sexually nor conjugate, and which cannot possibly intercross, uniformity of character, so them all staying the same, can be retained by them only through the principle of inheritance. So this is what's going on in your body, right? Your cells are derived from a single clone. They're inheriting and passing on genomes when they divide. And the way that they would stay the same is either from things not changing, which they do, or through natural selection, which will destroy individuals departing from the proper type. And I guess what I would encourage you to think about, especially if we're thinking about cause unknown chronic diseases, things like your dementias, is that the immune system can be a naturally selective force that decides what types of cells live and die. And obviously my thinking is very much in this direction from cancer immunotherapy, tumor immunology. If you can get the immune system to recognize a melanoma as foreign, it is very good at eliminating every single clone derived from those cells. And I assume that this is also part of the natural aging process and part of what's causing things like arthritis and dementia and osteoporosis and such with time. So what do we know about autoantibodies? So this is just these are called hits on Huprot microarrays. So obviously they're all human proteins, not mouse, but we see similar results in a mouse phage display assay that we run, which is that in general, I tend to see more autoimmune reactions on human samples than we see in research mice. One rather obvious explanation for this is that in a sheltered condition that are not exposed to viruses, not as exposed to different types of foods, mice are not having as many of these types of exposures, though they definitely do still have autoantibodies. They're just rarer than seem to happen in humans. So obviously we live in a little more dangerous outbred environment. I imagine that if we were catching wild mice, they might have a few more antibodies like humans do. This is now kind of getting back into an assay. So this is the same slide that I showed you before, just to kind of remind. So we make the arrays, you stain with serum, you come back with a secondary, you just see what lights up. And so this is what one of those physical slides looks like. So we have a lot of landmarks and immunoglobulins that the secondaries hit. So secondary hits quite a few things. This is now what a healthy human serum looks like. And if you kind of look closely, you'll see a couple of little dots that are starting to light up that are occurring over here on the right that aren't occurring on the left. And that's essentially what makes a plot like this, right? So two of those arrays over time, these are the dots that are lighting up that are not secondary specific. That's really what our data looks like. If someone has catastrophic autoimmunity, it looks a lot more like this off to the right. So this is a sample from an air knockout cohort that we've run in collaboration with Mihalis Lionikis at NIAID. And it shows that people or children that have lost their central tolerance, they have autoimmunity just widespread and catastrophic to most of the things on the chip. But this is still very unique. And each of the kids is going to have autoimmunity to about 8,000 of these random human proteins. And that's all going to be very unique to them. So most of the things that are shared, even in an APS1 or an air knockout patient type setting, where they're missing the air transcription factor in the thymus that is so essential to central tolerance, they're still getting very unique autoantibodies as well. And so now I'm going to share some results from a manuscript that we have in preparation on identical twin aging. So we've done this with Mario Falchi at Twins UK. And what we have here are sets of twin pairs before and after 15 to 22 years of aging. So a pretty long time gap. And they're all identical twins. They're all women. And it's from about age 55 to 70 plus. We also have obese and lean twin pairs. There wasn't as much of an effect with that. But so 80 total serum samples, 40 different people. That makes 20 pairs of twins before and after over a decade of aging. And so we ran Huprots on these people. So there's actually the same plots that I showed before. So everyone has, between 200 and 400 or so autoantibodies in the IgG channel, slightly fewer number in IgA. That might just be that the staining is a bit more optimized for IgG than IgA. So to kind of help you understand the results, so it's important to know what a Jacquard similarity is. It's a really simple statistical tool. It's essentially just a ratio of a Venn diagram. So how many responses are literally identical in both samples versus different. Obviously, so all of our hits that I'm doing and using in this plot are based on z-scores. So things that are not secondary only reactive, that also have a z-score greater than 3 within that individual array are called hits. If it's below a 3 threshold, it gets cut off. So even tech reps are going to lose some just from kind of some drift at the bottom of the list as to who's winning and who's in the list and who's not. So this is now data from these identical twin pairs. And so what these are the similarities of all the total responses. So what are the things that are lighting up on a Huprot chip at time point A versus time point B 15 years later? So to start, we have technical replicates over here. So you just run the same serum from the same time point twice. You get a similarity of about 0.8. It's a little bit lower for the IgA because the staining's a little bit dimmer. But everybody versus themselves is coming out around a 0.5 versus their twin being much lower and strangers being even lower than that. And essentially the twins looking a bit more like strangers than themselves before and after aging. Themselves before and after aging look a lot like tech reps. And so what this is showing is that the immune response and the autoantibodies that are occurring in these patients, both IgG and IgA autoantibodies, are highly unique to themselves, don't look anything like a twin, and very stable with time. And so I guess what I would challenge you to think about is if we're talking about things with autoantibodies that they can do that can permanently impact your health, like blocking a cytokine, blocking vitamin B12 import, there's probably a lot of health outcome that's built in here. And there's not a lot of that really being explored today. And I think we're a long way from really understanding what all this uniqueness means. This is just kind of another kind of refresher that even, a 0.5 or so is meaning that, over half of the responses are literally shared before and after. So it's looking a lot like a tech rep. versus age self, whereas a twin or a stranger is a smaller portion of the repertoire. Though there are public autoantibodies, that of course does occur. This is now the exact same samples and the exact same experiment, but two entirely different orthogonal platforms. This is both our VIR-scan and our HuScan phage display immunoprecipitation sequencing. So I mentioned this a little bit before. So we have these libraries of bacteriophages that are either expressing 500,000 chunks of pretty much every virus in UniProt, or 700,000 chunks of little peptides representing the human proteome. You then react serum samples with it. You do a protein AG pull down that's just kind of grabbing global IgG. You then barcode and sequence these samples. These little control plots are showing that the majority of the library is there in the sequencing assay, and that people are getting hits, and they're getting a lot more hits than a decoy set of samples. So just the protein AG beads without serum. So everyone's got large numbers of hits, and you see that there's quite a range to this. I don't know if I have a plot on it, but in general, people's total numbers of hits are pretty stable with time, not just the identities. This is now the result of the identities, and what you'll see are these plots look a lot like the ones that we showed before. So the antiviral repertoire is about 0.8. It's really, really stable with tech reps. You see it's very similar to HuProt, about 0.5 versus age self. Everyone looks more like their twin than a stranger, but they look more like strangers than their twin. And it's the same for both the antiviral and the anti-human autoantibodies. And I'd actually forgotten about this slide, but this is an actual separate peptide hormone phage display library, which we have made. So if you have an idea about a FIPSeq library, an antigen set that does not exist right now, that's actually something that we've acquired the ability and can do and build for you. So I created this library based on a database of human peptide hormones. So things like glucagon and insulin and vasoactive intestinal peptide, things that are these really critical tiny signal sequences that are made from pre-pro proteins that are in the larger HuScan library. And we made a bunch of different windows of them and variants and saw similar things in this. So people do have autoantibodies that bind to peptide hormones. We do maybe see a little more uniqueness shared with the twin, which I think is a little interesting, especially for things that are so critical. We've got some people that are being a bit more shared than others, and then the strangers as before. And so kind of putting it all together, so this is now all the different assays. So it's human proteome microarrays, both in IgG and IgA, the VIR-scan, the HuScan, and the peptide hormone library. Like all of them are essentially screaming the same thing, which is that everybody is really unique, a lot more similar to a tech rep than to their identical twin or a stranger. And we know this, right? So the adaptive immune system is based on, lots of acquired randomness, and there's a lot more that goes on to the creation of this repertoire than just shared environment, right? So there's a very strong individuality to all of this. But this is now just an example of one of these peptide hormones I was talking about. This is to insulin-like growth factor 1. And if we kind of look, so it's kind of hard to see because the lean and aged are kind of, you're hopping over here. But what you can see is that there are 5 of 5 versions of these that we're making that people are hitting at both time points. So we have lots of built-in replicates of these peptide hormones. There is a specific piece of this one that multiple people seem to be seeing that other people don't seem to see, and that this is relatively stable with time. And so responses like this are what create plots like that peptide hormone similarity over to the right. And this is a kind of a separate type of plots. This is what I was saying before, where generally you have new responses that come rather than old ones that go away. So there's a threshold for all of these assays where people are generally getting more total hits with time. So now ignoring kind of the uniqueness, you generally are seeing a few new things that are going on. So new viral infections, new autoimmune mistakes. So this kind of becomes cumulative across the patient's lifespan. And we also see similar things in mice. So this is now, I guess this is mouse scan data. So we have a mouse proteome phage display. So using long peptides, again, FIPSeq, react with mouse serum. These are now mouse proteins. And we're looking at a few different strains, either BALB-C, BLAC6, or CD1, which is an actual outbred strain, which has MHC diversity. And so we didn't have sequential time points on these. So really, we're just kind of comparing literal tech reps and to within strain versus other strains. So individual mice look like serum pools from the same strain, and they don't really look like the antibodies from the other strains. The similarities aren't that great for BALB and Black6 because the total numbers of hits were actually so low. The CD1 outbred animals seem to have quite a few more hits, so the similarities for the tech reps are now looking more like the human, and we saw the same effect with that, where they looked a little bit more like the other CD1s, or at least the public antibodies that were shared were more likely to be shared within strains than from the foreign strains, which is kind of a similar experiment to the identical twin people. We also ran the Q-prot arrays. This was the same data that I showed a little bit earlier, showing that some of these BALBs are like the BALBs are getting fewer than 20 or so hits on a Q-prot chip, whereas the CD1 females are having nearly 100. So quite a different range. And we see the same thing on the Q-prot chips that I just showed you on the literally murine phage display, which is that all the animals look a little bit more like individuals within strains than stranger strains. And kind of to throw a wrench in this, because I've mostly been talking about the uniqueness and stability of the repertoire, kind of the ending punchline is going to be that this is also an isotype-specific phenomenon. So we are only just starting to get into looking at this, but kind of the takeaway would be that IgG does not really exist. It's composed of IgG 1, 2, 3, and 4. They've got different Fc binding functions. They do different things. There's also IgE, IgD, IgM. And right now, almost all of this data that we gather is mashed together. And I think it kind of confuses people. And so one of the things that we're trying to build for the future is an actual pan-human isotype panel. So this is the result of screening thousands of hybridomas in our lab in Puerto Rico, where we've actually gone ahead and made a full panel for all the human isotypes right now, other than IgG2. And what I'd like to be able to see us doing in the future is, in addition to doing proteome-wide autoantibody surveys, to be able to do proteome-wide isotype-specific autoantibody surveys, because my guess is there's going to be data in the multi-isotype staining, just like there is on a flow panel with cells, right? Like if you're, depending on what markers you're positive for means different things. My guess is overlap to stuff like the CD4 and CD8 response might have to do with what's going on in the isotype code, which is not really deconvoluted yet. And to show a little bit of data where we've played with this, so this is using quite a few microarrays on just a few samples, where we're individually staining in the same channel with all of these different panels. This is before we built the whole panel. So this is IgM, total IgG, 3, 4, A, and E. And we're looking at all of that simultaneously in a single individual before and after 5 years of aging. We're not going to stay on this plot, but this is kind of what it looks like if you cross-compare all of them. So things like IgE, IgM dual positive, IgG3, IgE dual positive. So there's now kind of extra richness to this data. And if you kind of glance around, you'll see places where they correlate or where they anti-correlate and seem to avoid each other. And very little of this has really been looked at to date. And all I've got really is this one sample. And even just looking in the total isotypes, we can see some pretty clear things going on. So this is, again, before and after the COVID-19 pandemic. We see that IgA and IgG3 behave a lot like I'm used to seeing total IgG behaving, which is most of it stable across the axis. And there's a few new things, including a new response to COVID-19 protein. IgE is very different. You can imagine that it's pretty dangerous to have autoantibodies that are IgE. And it looks like that these mostly have not survived the 5 years of aging, and there's new ones that are occurring instead. IgG4 is a bit more interesting. So there were very few beforehand, and now there's quite a great many, and it's tracking with COVID-19 response. So there's a growing literature on people that are getting repeated mRNA vaccine injections due to that hanging around in a tolerogenic sterile environment for a long time, potentially inducing a tolerogenic cytokine like IgG4. And what's kind of nice and interesting about this group is there seems to be several IgG4 autoantibodies that are tracking with that. So I don't know if that's good or bad, but something that would be worth investigating, and I hope we get to do a bit more of in the future. Kind of one last thing. So, because as I promised, we were getting lists of all these pan or multi-isotype patterns, things that are either dual positive, single positive, positive for one, explicitly negative for another. So this is from this plot before that I was showing, where we have all these different quadrants, where I'm just kind of crudely calling dual positive or single positive while considering different groups. And I threw all of the things that were over here on the right that weren't here on the left. So we looked for the changes before and after aging and wanted to see, was there any shared features of this in ontology enrichment versus the Human Protein Atlas? And some things that we see right off the bat are things that we know track with autoantibodies. In general, you do any ontology enrichment for autoantibodies, you're going to get intracellular proteins. So we see lots of different multi-isotype patterns that are hitting either RNA or things that, things that are intracellular or nuclear. There's some that are a little more interesting, CD markers. You can imagine that that might be affecting immunity. And something that I've also learned about this patient since originally running the samples is that they have Hashimoto's thyroiditis. And we see quite a few of these that are going to thyroid category proteins. So there's something going on, I imagine, with that and with the patient's diagnosis with that condition in the interceding 5 years. And so kind of to leave you with, as we kind of start wrapping up, so a lot of this has been about autoantibodies or antiviral antibodies. There's a lot that antibodies can potentially be binding. It's essentially all of our experience as living organisms on Earth, both to other life we encounter, as well as artificial structures, which are now induced into our environment. And my guess is there's a lot of things that are non-toxic, that are non-mutagenic, things like microplastics that are hot in the news these days, that probably are not directly harmful to human cells, but might be chemically transforming proteins and inducing autoantibody responses. And I think there's a very fertile field of discovery for this type of stuff as we kind of proceed. And I imagine that we immunologists are going to have careers in this, regardless of how good the AI gets. At least for the near future. And so I guess with that, I would turn it back to Mike and the audience, and I can both go to the chat as well as if people want to just unmute and speak up. Happy to talk about anything else for the final 15 minutes.
Thanks, Tyler. Yeah, as Tyler mentioned, we can open things up for questions. So you have to unmute to ask a question, or you can put it in the chat and we can see what we have there. Yep.
Yeah, so I guess I'll start with the first one, which is, hi, Tyler, thanks for this amazing talk. Thinking about examples of unexpected autoantibodies raised against B12, maybe some vaccines. Do you think this could have implications leading to the development of cancers? I think so. So I guess let's back up for a second. So we're actually going to be putting in a grant with Professor Casanova following up on their infectious disease stuff. So dating back to when I was in graduate school, I was seeing people that were ipilimumab or nivolumab responders versus non-responders have anti-interferon family autoantibodies. I wasn't able to follow up on that. We had to give the data back to BMS. I didn't have the rights to publish on it, but it's kind of long been a hunch of mine that the reason that highly immunogenic tumors like melanoma are allowed to exist in the first place might be from these types of mistakes. So Casanova has shown in tens of thousands of samples that about 5% of people over age 65 have interferon alpha knockout-like autoantibodies. And my guess is this most definitely does have to do with tumor incidence. There were probably other things, either related to natural immunosurveillance or metabolic issues, which are brought up that I imagine are coming from. Where the B12 one comes from in particular is not really clear. So it doesn't seem to have a good homolog or mimic with viruses, though yeast and other commensals do have transcobalamin in them. And so I wouldn't be surprised if it came from something like that. So I guess the literature is to some degree extrapolative at this point, though there is a really nice paper that we helped with by Aman Osman's group with people that are getting ipilimumab, nivolumab. There's two different papers, and they have to do with differential immune-related adverse events as well as outcomes. So there are things in the autoantibodies that seem to predict whether or not someone's going to be an IPI or an anti-PD-1 or an anti-CTLA-4 responder before they get the therapy. And I can tell you that some of those things are to cytokines and or cytokine receptors. They don't explicitly listed in the paper for some reason, though I think some of them are in a patent they submitted.
Are there other questions?
Christian Moritz, great. Yeah, so we don't natively put COVID-19 spots on the chip, though we do have a spike and a nucleocapsid that are from the original Wuhan strain that we will put on it upon request, just like a standard custom HuProt spot. So if you'd like that in an upcoming study, reach out and tell us about it, and we'd be happy to include that. Long term, I'd really like to see the majority, because like a lot of people that are running our VirScan assay, a lot of them just want to know simple exposure. And there's a lot of vaccine and viral antigens that we can just add things like that to the HuProt chip. And I'd really like to see that in a future, quote unquote, HuProt 5.0 as a standard.
And as Tyler mentioned, so the HuProt array has 21,000 full-length recombinant human proteins on it, but we have the ability to add custom content. So it's just a small fee to add up to 10 additional spots or 10 additional antigens to the array.
Damien?
Hi, Tyler. I really like the presentation. Tell me, what do you think about the similarity index that you got when you were running replicate, right? You were getting an 0.8, which I think it's pretty great given the small volume, right? You're capturing versus the total possibilities, right? So what do you think about that? How many replicates? How robust is that? Because I'm impressed it reaches that high.
Yeah, so a lot of that is defined by the fact that you have to binarize the data. You have to call a false or a positive to do that type of assay. And so by binarizing it, there's essentially the dimmer signals that are just above the threshold. They're competing with each other, and their order is swapping around. And I guess that's kind of what I was showing with the. So if you showed a scatter plot, it would actually look even better than that to some degree. But yeah, so we're surprisingly reproducible. So there's several things that affect HuProt reproducibility. It's like the lot of the print lot of the proteins, as well as the manual skill of the operator. And we have quite a few QC tools where we're actually manually checking the top outlier spots in every array stained in our lab in Mayaguez. Pedro and his team are really exceptional at that, where if you've ever run a microarray, like dye artifacts are a pretty common thing that affect data. But we're really good at eliminating that, and that 0.8 is very much a testament to the quality of the staining that we have. And so that's either something we're able to do for you guys as a service, or we'll actually fly Pedro and Oscar and our staining team out to help set up core facilities. So if that's something you're interested in doing as well at UPenn, that's something we can help you with.